Welcome to part 4 of this video on the immune system. Okay, so I'd like to begin this video with a summary of where we've got to so far. So we had this pathogen which had breached the first line of defense and had got into one of the tissues of our body. We then saw that its presence would be recognized by sentinel cells, so mast cells, macrophages, and dendritic cells. They would use their variety of pattern recognition receptors to recognize molecules associated with the presence of the pathogen, PAMPs. Okay, what they would then do is set off the alarm by releasing a whole bunch of pro-inflammatory mediators, and we've got the list of 6 pro-inflammatory mediators here. So far, what we have discussed is the roles of 4 of these pro-inflammatory mediators. So we've discussed that histamine, released solely by the mast cells, is going to be the first one to actually cause the acute inflammatory response. It's going to be released extremely rapidly, and it's going to act on the endothelial cells of the microvasculature extremely rapidly, and it's going to lead to type 1 activation of those endothelial cells, and we've seen the effects of type 1 activation. It leads to the endothelial cells producing prostaglandin E2, which will cause vasodilatation of the terminal arterioles and therefore increased blood flow to the affected area. And we discussed the reason that you would want that, which is that all the troops for destroying the pathogen are present within the bloodstream, and therefore if we're going to recruit them into the extracellular fluid, then having an increased blood flow means that we're going to have an increased opportunity to recruit the troops. We're also going to undergo endothelial contraction in response to type 1 activation, which will allow the formation of an exudate, which will contain lots of the proteins from the blood, which will be capable of attacking the pathogen directly. And we're also going to start recruiting leukocytes, white blood cells, from the bloodstream, which will be able to attack the pathogen as well. And specifically, after type 1 activation, the endothelial cells of the capillaries and postcapillary venules will recruit neutrophils. We've then seen the role of tumor necrosis factor alpha and interleukin-1. Now remember, they act after histamine. Histamine acts quickest. TNF-alpha and interleukin-1, they have to be synthesized first, and that protein synthesis process is a time-consuming process. So it takes time for the sentinel cells to actually synthesize those molecules. In addition, when they actually act on the endothelial cells, the signaling pathway takes longer to actually take effect. Type 2 activation, which is the one induced by tumor necrosis factor alpha and interleukin-1, it takes longer. It requires changes in gene expression within the endothelial cell to elicit the changes in behavior, whereas type 1 activation didn't. It didn't require any new proteins to be synthesized. And we've discussed that type 2 activation is just going to do all the things type 1 activation does, but does it better. So you get massive production of prostacyclin, causing massive increases in blood flow. You get endothelial cell remodeling of the cytoskeleton to cause much bigger gaps to be opened up between the endothelial cells, and therefore more exudate will form. You also can start recruiting even more neutrophils, and you can also start recruiting other types of leukocytes, for instance monocytes, which will differentiate into macrophages. Okay, we also saw what prostaglandin E2 secreted by the sentinel cells will do. That will do exactly the same thing as all this prostaglandin E2, which has been secreted by the endothelial cells that have been acted upon by histamine or TNF-alpha and interleukin-1 will do. That'll cause vasodilatation of the terminal arterioles and therefore an increase in blood flow. Okay, so we've seen the major components then of the acute inflammatory response. Where are we going to go from now? Well, we still have 2 more pro-inflammatory mediators to talk about, interleukin-6 and prostaglandin E2. I want to talk about what they are going to do. I also want to then go on to talking about what's the point of all of this. So I want to talk about what the exudate is actually going to do. So we'll talk about the complement system and all of the 3 different complement pathways. And then I want to talk about leukocyte extravasation more. So I firstly want to talk about the actual process of leukocyte extravasation, and then I want to talk about what the leukocytes, once they're in the tissue fluid, so the neutrophils and the macrophages, what are they actually going to do now to help destroy the pathogen? And that will finish our discussion of the acute inflammatory response. Okay, so that's the plan from here on in. So we'll start off with what does interleukin 6 do, and then we'll talk about what prostaglandin E2 is going to do. And broadly, interleukin 6 is responsible for something called the acute phase response and also giving you a fever, and prostaglandin E2 is going to be hugely involved in producing pain at the site of inflammation. Okay, so just getting this straight, let's begin then with interleukin 6. Okay, so the sentinel cells produce interleukin 6 when they sense that there is a pathogen present within the tissue. What is interleukin 6 going to do? Well, interleukin 6 is going to end up going into the bloodstream and it's going to cause systemic responses. Okay, so whole body responses to this infection that is present in one of the tissues. So it doesn't cause a local response at the site where the infection is actually occurring. It's going to cause a systemic response. It's going to make you, well, give you the signs that you are actually unwell. Okay, so firstly, I want to talk about something called the acute phase response, which is initiated by interleukin 6, which is a response of the liver to interleukin 6. And it's a response that is going to occur when we've got an infection. So this is my very simple picture of the liver here. And interleukin 6 is going to cause a response in the liver known as the acute phase response. Now, what is meant by the acute phase response? Well, acute means that it's going to begin very rapidly, and in fact it's going to arise when you have this infection and when you have the appearance of interleukin 6. And the response of the liver is going to be to synthesize loads more proteins that are involved in attacking pathogens. So I've discussed about the fact that the reason we want to form an inflammatory exudate is to bring in all sorts of proteins that are present in an inactive form in the bloodstream, but which are capable of attacking pathogens if you bring them into the tissue fluid where pathogens are present. Those proteins in the bloodstream, they are synthesized by the liver. The complement proteins are the principal ones that we're interested in here. These complement proteins are synthesized in the liver. If we're now going to suddenly take loads of them out of the bloodstream to use against the pathogens, then the liver's response needs to be to increase its production of them to make sure that we still have enough of them. Okay, so that's what the acute phase response is all about. The liver starts producing more of these proteins that it secretes into the bloodstream, which are involved in attacking pathogens. And as I say, the principal ones that we're talking about here are the complement proteins, and we will talk a lot about the complement proteins and the complement pathways later on when we come on to discuss what is the point of forming an exudate. So it is interleukin 6 which tells the liver that there is an infection present and that therefore we're going to be using a lot of the complement proteins, so could you please make some more? And it drives the acute phase response. So interleukin 6, secreted by the sentinel cells at the site of infection, will go into the bloodstream, go to the hepatocytes, act on receptors on the surface of the hepatocytes, and trigger intracellular cascades, which will lead to them producing more of certain proteins that they will secrete into the bloodstream. And these proteins are those proteins that are involved in attacking pathogens, such as the complement proteins. There are others beyond the complement proteins, but if you know the complement proteins, you have your answer as to a major class of proteins that the liver is going to start producing very rapidly in the acute phase response. Okay, so that's one of interleukin 6's functions. Another function is that it's going to cause a fever. So it's going to cause the raised core body temperature. And the reason that you get a fever when you are suffering with an infection is to try and make the environment more hostile for the growth of the pathogen. The idea is that if you raise body temperature, it might be the case that the pathogen is no longer able to thrive at this higher temperature, and therefore you might be able to control it in that way. So how do you actually bring about a fever? How do you raise core body temperature? Well, there is a portion of the hypothalamus that is responsible for controlling your body temperature. So let me just draw a little picture of the hypothalamus. So the hypothalamus is a portion of the brain that sits in front of and below the thalami. and I'll just draw a little picture of it here. So this is the optic chiasm that we're seeing here. So I'm drawing a picture from the left hand side of the hypothalamus. So we're looking from the left hand side, and here is the pituitary stalk with the pituitary gland dangling down here, and then there are two little processes dangling down from the hypothalamus at the back, one on the left, we're seeing the left one because we're looking from the left hand side, and one on the right, and they're known as the mammillary bodies, and they're shown there. So roughly this is what the hypothalamus looks like if you look at it from the left hand side. The thalami would be here, and the midbrain would be sort of here, and then the rest of the brainstem would go down here, and then you'd have the cerebral hemispheres above it. Okay, so all of this is the hypothalamus, and these are the mammillary bodies of the hypothalamus at the back there, like so. So I'll just colour it in green. And then you've got the pituitary stalk with the anterior and posterior pituitary glands here, and the optic chiasm just shown there. Okay, right, so there are two nuclei known as the anterior hypothalamic nuclei, one on both sides of the hypothalamus. So there is a left side of the hypothalamus and there is a right side of the hypothalamus, and if you're familiar with neuroanatomy you'll know that in between the two sides of the hypothalamus you've got the third ventricle, and you have these two anterior hypothalamic nuclei on either side of the third ventricle, and roughly on this picture we will see the left hypothalamic nucleus at this sort of position here. Okay, left anterior hypothalamic nucleus rather. So this is representing the anterior hypothalamic nucleus on the left side here. So the anterior hypothalamic nucleus, and I'll just put a little L there for left. And if we were looking at the right hand side we'd see the right anterior hypothalamic nucleus in this sort of position here. Now this nucleus is extremely important for controlling body temperature. So interleukin 6 is going to go up to this and these anterior hypothalamic nuclei, and it's going to reprogram them, basically reset them. It's going to say, okay, there's an infection present, can you raise core body temperature? And the left anterior hypothalamic nucleus will take all sorts of actions on the peripheral body to change the amount of heat that you're producing and the amount that you're losing, to shift the equilibrium so that core body temperature will rise. So this will now cause a fever, which is a raised core body temperature. Okay, so interleukin 6 is responsible for these two systemic responses to having an infection, the acute phase response and also a fever. Okay, right, so that's interleukin 6. Now what I want to talk about is prostaglandin E2. And this is going to be involved in making the area that is infected and now inflamed, because we brought in all of this exudate and these leukocytes. It's going to be involved in making that area painful. So let me now discuss how prostaglandin E2 is going to make the affected area painful. So in the tissues of your body you have what are known as nociceptors, which are attached to primary nociceptive afferent neurons. Let me just draw a little picture here. So let's just say that the tissue we're talking about is the skin, to give a nice example. So I'll just draw a little picture of the skin here. So there's the epidermis, and here is the dermis of the skin here, and then you'd have the fat layer underneath. Okay, now if we've got our pathogen present in the dermis here, so here's our pathogen, I'll just colour it in red there. And let's say that this is the area then that is undergoing the acute inflammatory response. Why is that area going to become painful? Well, prostaglandin E2 is going to be hugely involved in that. Now, this area of tissue will have a lot of structures known as nociceptors, and I want to draw a nociceptor on here. A nociceptor is basically the terminal peripheral end of a pain neuron, a primary pain neuron. So if I just draw this on here, so this would be roughly what a nociceptor looks like. It would look like the branches of a tree here, and it's just a structure at the end of a primary nociceptive afferent. So here is then the peripheral process of the primary nociceptive afferent, and what will happen is this axon will go into some nerve, and then it will make its way back up to the spinal cord, and the cell body of this neuron will be in the dorsal root ganglion of the spinal cord, and then its central process will enter the spinal cord. So if I just draw a quick picture of this, so if I just draw a quick picture of the spinal cord here, so here is the ventral median fissure there, and here is the dorsal median sulcus of the spinal cord. And if I just now put on the grey matter, like so, try and get it roughly symmetric. So just to give you a bit of the anatomy of the spinal cord, then this is the ventral median fissure. So we've taken a transverse cross-section of the spinal cord at the level that this neuron is going to be entering the spinal cord. This is the dorsal median sulcus at the back here. So dorsal median sulcus. And then we've got the grey matter and the white matter of the spinal cord. So I'll just colour this in. So we'll have, this is the grey matter of the spinal cord here in blue. And of course, the grey matter of the spinal cord kind of is in this butterfly shape when you take a transverse cross-section in this way. And these are known as the ventral horns of grey matter. So you have the right ventral horn and the left ventral horn here. And these are known as the dorsal horns of the grey matter. So the ventral horns will of course be the place where the cell bodies of the alpha motor neurons are present, and the dorsal horn is where the sensory neurons that are involved in pain are going to send their central process to synapse onto a second order neurons. And of course you will be aware that in the central nervous system, white matter represents axons and grey matter represents the cell bodies. So this is the dorsal median sulcus, ventral median fissure. Okay, so just completing this picture up, and let's say that we took this cross-section as an ideal level that we actually can see some roots coming off. So here is the dorsal root, and here is the ventral root. So the ventral root is where the axons of the alpha motor neurons and also any sympathetic or parasympathetic fibers that are coming out will come out. And the dorsal horn is the dorsal root here is where fibers will be entering into the spinal cord. And of course, you have a dorsal root ganglion present here, like so. So this is now my picture, and I've just gone a little bit over there, so I have to loop round. So here is my nociceptor. The nociceptor is the name for the sensory apparatus at the end of this pain neuron. So this tree structure here, this is the nociceptor. So if I just colour this in, this apparatus at the end of the neuron which detects noxious stimuli, this is what is meant by the nociceptor. So all of this portion, it's a structure in its own right at the end of this neuron. Okay, and all sorts of noxious stimuli. So a noxious stimulus is a stimulus capable of causing pain. And prostaglandin E2 is going to be indeed a noxious stimulus. But you know, you can come up with loads of other examples of noxious stimuli. So for instance, far too high temperatures would be a noxious stimuli. Far too low temperatures would be a noxious stimuli. Acidic pHs would be a noxious stimuli. At too high pressure applied to the skin would be a noxious stimuli. So there are all sorts of proteins in the membrane of these nociceptors which are capable of detecting many different noxious stimuli, and when those noxious stimuli activate those proteins, it will result in depolarization of the electrical potential difference across the cell membrane of the nociceptor. And then if all of the different depolarizations coming in from all over the nociceptor are great enough, then it will cause an action potential to fire along the axon of this neuron here that has the nociceptor on its end. And a neuron that has a nociceptor on its end is known as a primary nociceptive, so that means pertaining to pain, afferent. Primary nociceptive afferent. So afferent just means it's coming into the spinal cord. Nociceptive means it's relaying pain information. Primary means it's the first neuron along in the neural circuitry. So primary nociceptive afferent. Okay, and this primary nociceptive afferent, it will come in like so, and then it will have its cell body up here in the dorsal root ganglion, and then it will have another process coming off like so, which will go into the dorsal horn of the spinal cord here and synapse onto the second order nociceptive neuron, which will take the information up to the brain. So all of this is the primary nociceptive afferents, and primary nociceptive afferents are known as pseudo-unipolar neurons. So this is the description of the type of neuron that we're dealing with here. It's known as a pseudo-unipolar neuron or a pseudo-unipolar nerve cell. And the reason it's called that is unipolar means that there is a single process coming off the cell body, like so. So here's the cell body. You can see that there's a single process coming off. The reason it's called pseudo-unipolar is effectively it's a bipolar neuron with two processes. So even though there's only one process coming off the cell body, you can see that this very quickly splits into two separate processes. One that goes into the central nervous system, this one here, known as the central process. Okay, so I'll just highlight this in. So this one here is the central process, and then the other one that's going out to connect up to the nociceptor, which is known as the peripheral process. And I'll highlight that one up as well. We'll have that one in, I think, yellow. So this is the peripheral process. So it's described as a pseudo-unipolar neuron. Right, so to summarize what we've got to so far, tissues are innervated by primary nociceptive afferents, which have on their end these nociceptor apparatus, which have in their cell membranes, they're these tree-like structures, and they're covered in proteins that are capable of detecting noxious stimuli. And by the way, nociceptor, if you ever hear someone talking about the concept of a nociceptor, that's the name for the actual proteins in the membrane of the nociceptor. So the nociceptor is this entire structure here. A nociceptor is one of the proteins in the cell membrane of the nociceptor, which is capable of detecting noxious stimuli. So actually my flow diagram should be like this. Noxious stimuli activates nociceptor. Nociceptor will depolarize the membrane of the nociceptor once it's been activated, and that all of these depolarizations coming from all over the nociceptor can sum together to actually cause an action potential to fire up the peripheral process of the primary nociceptive afferent, and it will then go down the central process and cause the release of neurotransmitter, which will then activate the second order neuron, which I'll just draw here, which will have its cell body in the dorsal horn of the spinal cord, and its axon will then cross the midline and ascend in the anterolateral ascending white matter column up to the brain. So they'll relay the information up to the brain, and this is what causes pain. So when these neurons are activated, they relay messages up to the brain, and that then causes the conscious experience of pain at that area where the primary nociceptive afferents were activated. Okay, so that's just a little bit of a review of how pain works. Now let's talk about what prostaglandin E2 is going to do. So if we start producing lots of prostaglandin E2, what it does is it works on the nociceptors in that area. It works on receptors in the membranes of the nociceptors, and it causes loads of different things that are going to lead to the activation of the nociceptors. But one of the current viewpoints, and it's not completely established, but I think it's growing in how generally accepted it is. One of the newer ideas about how prostaglandin E2 actually causes pain in the affected area is that it causes changes inside the nociceptor, which are then going to lead to spontaneous and repeated, or rather, what's the better word? Repeated is the good word. Repeated activation of the rhythmic, maybe is a better word. Rhythmic activation of the nociceptor. So it's actually very similar to the heart. In the sinoatrial node cells of the heart, which you will know continually generate electrical signals on a rhythmic basis, there are special channels known as HCN channels. So HCN channels are the special channels in the cells of the sinoatrial node of the heart, and these are involved in creating rhythmic electrical activity, creating action potentials on a rhythmic basis. And what's believed to actually happen is when prostaglandin E2 stimulates the nociceptors, it's believed to actually activate these HCN channels, and they will now start setting up electrical activity on a rhythmic basis in these nociceptors and causing action potentials to fire up the primary nociceptive afferents on a rhythmic basis. So you're going to get action potential after action potential after action potential after action potential after action potential on a rhythmic basis once the prostaglandin E2 has actually stimulated the nociceptor, and it's believed to be because the prostaglandin E2 results in intracellular cascades which are going to activate the HCN channels. And remember, these HCN channels, the thing that allows them to actually generate action potentials on a rhythmic basis is that unlike most voltage-gated ion channels, which are activated by depolarization, these ones are activated by hyperpolarization. So when the electrical potential difference gets to the resting level, they will open and then depolarize the electrical potential difference up to threshold potential, and then the more normal voltage-gated sodium channels will then open and trigger the normal action potential. So that's, remember, what allows them to generate action potentials on a rhythmic basis, because the instant the action potential ends, you'll get hyperpolarization of the electrical potential difference again, and they'll then open. So that's the way in which they can generate spontaneous, repeated, rhythmic electrical activity in cells. And indeed, we think prostaglandin E2, by activating these, is going to then lead to rhythmic generation of action potentials firing along these primary nociceptive afferents, and that will lead to the continuous pain that you feel in an area that is suffering with an infection. Okay, so that's what prostaglandin E2 does. So to summarize now, we've seen how all of the cardinal signs of inflammation are produced. Remember that 4 major cardinal signs of inflammation are redness, heat, swelling, and pain. The redness and heat will be produced because of the increased blood flow to the area, and we've seen how that's produced. The swelling will be produced by the inflammatory exudate and also the recruitment of all of those leukocytes into the affected area tissue fluid. And the pain will be produced by the prostaglandin E2 here. So I think we're going to have a break here, and in the next video what we will do is we'll start actually looking at what is the purpose of forming an inflammatory exudate. So we'll look at the complement system.